All right. Hello, everyone. Welcome to our climate resilience and mitigation speaker series. I'm really happy to bring to you a new speaker coming to us from the East Coast. Even though her background is in Washington State, Yaaminin Venkataraman is actually was a grad student who recently finished at UW in Seattle, and she's now a postdoctoral scholar at Woods Hole Oceanographic over on the East Coast. So hopefully the weather is not quite as shitty as it is right here on the West Coast at the moment. So, so great to have you today. Just want to let everybody know that these are being recorded. So if you miss some of this, have to leave early, we will have these available. I'll send it out with the next go round, and all the talks are recorded, so you can check those out at any time when you get a chance, or share them with other folks would be lovely. Just want to give our speaker as much time as she needs. So I'm going to go ahead and introduce her, and then she can take it away. So Dr. Venkataraman is a recent PhD graduate from the UW School of Aquatic and Fishery Sciences. Originally from California Bay Area, she grew up going to the Monterey Bay Aquarium. Those visits sparked her interest in understanding how complex environments can shape organisms themselves. Since then, she's studied several different species environment interactions, including copepod and copper toxicity in tide pools, and limpet surf grass interactions under acidified conditions at UC San Diego, where she received her BS in general biology and a BA in environmental policy. For a PhD with Dr. Stephen Roberts, she used molecular techniques, Omics, to explore how Pacific oysters respond to changes in their environment. She now examines how other marine invertebrates, invasive European green crabs, and Caribbean corals rapidly respond to climate stressors on a molecular level in her new job as postdoctoral scholar at WHOI. Aside from research, Yaaminin is actively involved in science communication, outreach, and justice, equity, diversity, and inclusion efforts. Yaaminin aspires to be a professor who uplifts and empowers her students and community in the future. So great to have you, Dr. Venkataraman, and it's all yours.
All right, thank you. And thank you all for having me virtually. Like Dave mentioned, I am now at Woods Hole Oceanographic Institution, which is the historical and current lands of the Wampanoag tribes. And I am very grateful to be able to live here and work here, and hopefully can contribute to the tribes and be an active community member for them as well. So my title today is Oysters, Ocean Acidification, and Omics. And I picked it for two reasons. One, because it accurately describes my work, and two, because it's a fun alliteration. And all three are very interconnected, and that is what I hope to show you today. So let's move forward. There we go. So we are, as I'm sure you are all aware in this class, living in an era of rapid environmental change. So in Washington and the Pacific Northwest broadly, our canary in a coal mine was oysters in a hatchery. So in 2012, shellfish scientists and hatchery operators at the Whiskey Creek Hatchery learned that upwelled waters brought into the hatchery were naturally acidic and affecting their oysters' growth. So since then, ocean acidification has been a focus of hatchery and resource manager policies, adaptation strategies, and a lot of fodder for researchers who are really interested in learning how climate change impacts these really important organisms. So oysters, I like to think about oysters in three different ways. They are ecologically, economically, and socially important animals. So oysters, ecologically, are what we call ecosystem engineers. So when they're out in bays or along the seashore, they actually clump up on top of each other, forming these oyster reefs. And oyster reefs protect our shorelines from crumbling away in stormy weather, which I'm sure you all are experiencing in abundance right now. Oysters also filter feed, which impacts water clarity, so it improves it. And that can increase the amount of light that penetrates the water and promotes growth of eelgrass beds or other aquatic vegetation. In 2013, Washington State shellfish farms owed over a third of their profits, so about $25 million, to oysters. And especially in coastal counties, shellfish hatcheries and farms are often really large employers and contribute a lot to the state economy. And finally, we can think about oysters and shellfish more broadly. There's a social importance, especially in tribal communities. There are often customs around harvesting and consuming traditional food resources like clams. But shellfish and oysters are threatened by climate change and specifically ocean acidification. So scientists have monitored atmospheric and oceanic carbon dioxide concentrations at Mauna Loa Observatory in Hawaii. And over time, they've seen atmospheric carbon dioxide has increased, which then gets absorbed by the ocean. It is the biggest carbon sink. And then that decreases ocean pH. So over time, the ocean has gotten about 25% more acidic, which is categorized as a 0.1 unit decrease in pH. And these changes are often even worse in coastal or estuarine regions like Puget Sound, because there's differences in the oceanography and mixing and how those bays are interconnected. So when we think about ocean acidification impacts on oysters, there are two main categories. One, in larvae or these young oysters, we see developmental delays, which may be associated with like reduced shell growth and poorer survival. Oysters also undergo this metamorphosis phase where they transition from these larvae that I have up on the screen to oyster seed or spat. So they go from being these larvae that swim in the kind of like are at the mercy of the tides, and they are swimming in the water column, more or less. And then you have oysters that we know today, like that you may see like adult oysters where they're just kind of like stuck on to the ground, they're not moving. And so during that transition phase, exposure to acidic conditions can reduce the amount of energy these oysters produce, which makes it harder for that metamorphosis process to occur. We also see difficulty in calcification or shell building processes in adulthood. And perhaps an oyster survives as a larvae just fine, and it's able to grow its shell in these acidic conditions, but ocean acidification can hamper reproduction. So oysters have a limited amount of energy that they have to budget out to important things like getting food, building their shell, and reproduction. And if they're spending too much of that energy budget coping with stressors, there's not nearly enough energy left for them to develop sperm or eggs effectively. So changes in gamete development can occur in these acidified, stressful conditions, and that can also lead to differences in fertilization success. So we may not be able to have these like next generations of oysters that are as healthy as they would be if they were in non-acidic conditions. So if we think broadly then about how ocean acidification acts on oysters, we can think of it in the terms of like natural selection. So ocean acidification is this big stressor that acts as a selection event, and it removes oysters that are less tolerant of the stressor from the environment. So on the left here, I've got a bunch of oysters of different colors, and these blue and green and pink ones maybe aren't as great at dealing with ocean acidification. So they get removed from the population, they die. And now we're left with these purple and orange oysters. And while that can tell us something about how oysters respond to ocean acidification, there's still so much that this doesn't cover. For example, it doesn't really account for any non-lethal effects of ocean acidification on oysters, so any effects that wouldn't lead to them dying, or effects that aren't easily observable, like mortality or reduced shell growth. And we also have a limited understanding of why we see certain responses to ocean acidification. What are the mechanisms behind these responses? So while this framing is great, we need a way to understand the connection between the environment, so ocean acidification conditions, and the oyster at a molecular level, so we can investigate impacts we can't see, understand mechanisms, and then apply them to resilient aquaculture industries, right? At the very end of the day, we want to make sure we can keep oyster production in coastal communities up so that it doesn't disrupt food systems, people are employed. It's kind of important. So for my dissertation at UW, I leveraged omic or molecular techniques to understand how oysters respond to ocean acidification. And then using these molecular methods, I could untangle what is sometimes a really complicated web of ocean acidification effects on oysters and understand processes we wouldn't be able to understand otherwise. Right, so what do I mean by omics or molecular techniques? So we'd start with DNA, right? It's the DNA is kind of like a cookbook that holds all of these different recipes for life, right? The recipes or genes are then transcribed to a different molecule called RNA, and this is kind of akin to assembling all the ingredients that you're going to need to make a recipe. Then those RNA molecules are translated into proteins, right? The food. Proteins are the doers of the cell, and they're responsible for basically everything that happens within a living organism. So today, I'm going to start by talking about some of the proteomic work I've done in the past, which is the study, again, of all proteins in an organism and their functions. And then I'll talk about some of my work on the DNA side. So we'll start with proteins and then go to DNA. So for some of my work, I was really interested in how pH variation in a natural environment could drive differential protein abundance in oysters, with the idea that we can look at these differences in protein abundance to tell us how an oyster may be responding to ocean acidification on that non-lethal level, right? In a way that it's not dying, but clearly something in the environment is different than what would be ideal. So how is it responding and how is it able to maintain itself? To do that, I partnered with the Department of Natural Resources and some scientists at the UW Biology Department, and we outplanted oysters in 4 different sites in Puget Sound, Hidalgo Bay, Port Gamble Bay, the Skokomish River Delta and Case Inlet, and then we had another site down at Willapa Bay. And these are all areas where oysters are grown commercially. And we put them in 5 different sites and then 2 different habitats, eelgrass beds and non-eelgrass beds. So eelgrass beds are these are it's aquatic vegetation, and they are kind of like plants. So they'll every time they photosynthesize, they could increase the, every time they photosynthesize and respire, they can change the pH chemistry in the water. So we thought that maybe putting some oysters near eelgrass may be beneficial to them. Right, so we were hypothesizing that this local environmental conditions at each of these sites and eelgrass presence would drive these differential protein abundances. Right, so to recap, 150 sibling Pacific oysters were outplanted, 5 different sites, 2 different habitats, and at each of these sites we monitored pH, dissolved oxygen, salinity, and temperature. And at the very end of the month, we collected conidia or oyster gill tissue to examine all the different proteins in those tissues. So we can examine protein expression with 2 separate mass spectrometry methods, shotgun proteomics and selected reaction monitoring. So shotgun proteomics is more of a discovery phase. It allows us to answer the question, what proteins are there? And it provides us with this panorama picture. We can then use our shotgun proteomics results to inform a second method, which is called selection reaction monitoring or target analysis. So this is a method that allows us to zoom into what we're actually interested in, like specific environmental response proteins and how their expression varies. This gives us a very complicated web of data, which we don't need to talk about, but I want to tell you about the takeaways. So our takeaways were that 1, peptide or protein abundances were different in Willapa Bay compared to all the other Puget Sound sites. There were 6 proteins at Willapa Bay that had higher abundance than those Puget Sound sites. And it wasn't pH that drove those differences. It was warmer, less variable temperatures at Willapa Bay that influenced those abundances. So once we know that there's a difference, we can look into what those proteins actually do, because that's going to give us the meat of the information that we want to know. So what do these proteins do? We had 2 different categories of proteins. The first are antioxidant enzymes, and these are response to something called reactive oxygen species, or ROS. Reactive oxygen species are things that are produced anytime your cells are functioning, right? These are produced kind of as waste products, and it's really important that they're taken care of, otherwise they can damage cells. So increases in these reactive oxygen species, or reactive oxidative species, these antioxidant enzymes that target these ROS are important because it tells us that we're increasing abundance of these antioxidant enzymes to maintain our metabolic activity in a stressful condition. So they're trying to keep going and keep maintaining themselves, even though there's warmer and less variable temperatures in this different Willapa Bay area. And there was another category of proteins called molecular chaperones. And these are more of a general response. So a molecular chaperone is a protein that helps another protein refold itself. Proteins have to be folded correctly for them to work. So there's all these molecular chaperones that make sure all the proteins are folded correctly. So so what we can think about then is maybe this stress of temperature was misshaping some proteins, and there needed to be more molecular chaperones to reshape them properly. And this is what we learned when we looked at these proteomics, right? These oysters didn't really die, but they were placed in all these different locations. They still survived, but these oysters at Willapa Bay were responding to a very different set of conditions, specifically temperature conditions. And looking at proteins helps us kind of unpack what's happening at each of these locations. So now that we know we can use omic techniques to look at these sub and non-lethal effects of environmental conditions, and then we can say, hey, in Willapa Bay, these oysters are maybe experiencing more stressful temperatures. Aquaculture operations can then account for that as they account for that in their growing processes. So it's important information to have if you've got an oyster farm in Willapa Bay. So what about mechanisms for resilience in oysters? How can we connect environments with oyster physiology? So I've mentioned that I've worked with DNA before. And again, I've mentioned that you can think of DNA like a cookbook, where you've got this giant cookbook and all these individual genes are recipes. So we actually now know that changes in the environment, like ocean acidification, can change some of these recipes in a way. What happens is a stress like ocean acidification can add or subtract these kind of orange markers on DNA. And these are what are called methyl groups. These are just like little things attached to the DNA. The marks don't change the DNA sequence itself, but they can change how genes are interpreted or expressed. And this is something called DNA methylation. Again, right, if we think of DNA as a recipe, or DNA as a cookbook and genes as our recipes, changes in the environment can maybe highlight certain passages in a recipe or cross them out and cover them in whiteout, right? That doesn't really change, like a presence of whiteout or highlighting doesn't change the recipe themselves, but it could change how you interpret it if you're reading a recipe. And that's kind of what DNA methylation does. It doesn't change the recipe, it doesn't change the DNA sequence, but it could change how we interpret it. And that could change gene expression and gives us a direct link between the environment and oyster physiology. And if we examine changes in methylation, that could tell us if oysters are able to cope with these rapidly changing conditions. So a lot of methylation research has actually been done in vertebrates like fishes. So one study examined the influence of temperature on European sea bass sex determination. And what they found was that in warmer temperatures, you had this change in methylation, which affected a single gene that then made it that you would have more males in a population over females. So a change in DNA methylation can really impact, like a change in DNA methylation associated with a change in the environment can really impact an organism on a pretty big level, which is why we're interested in studying it. So part of my work then was looking at how ocean acidification influenced these DNA methylation marks in female oysters and in their reproductive tissue, because reproduction is a really important process for oysters, and it's really important for hatchery managers and growers to kind of manipulate reproduction processes in a way where they're going to get the best oyster yield, right? You want to make sure you can have a lot of oysters. So they're really interested in how to kind of optimize these processes, and understanding all the little things that go into it is really important, especially in stressful conditions. So what I did was I exposed oysters to low pH stress for 7 weeks. And then we took their reproductive tissue, we sequenced the DNA in that tissue, and then identified different methylation patterns in those tissues based off of these oysters had seen low pH or ambient pH conditions. So if they were in stressful conditions or not stressful conditions. And what we found was that there were methylation patterns associated with some genes, right? Again, those individual recipes that could then impact the oyster as it developed and grew in low pH conditions. So what do these genes with those different methylation patterns do? Well, we found one group of genes associated with gonad or reproductive tissue development, specifically involved in oocyte or female egg maturation. So what this tells us, right, is that there are genes that have these different patterns in low pH conditions that are involved in producing eggs. And they're also involved in stress response and producing eggs. So if we take all that information together, it kind of tells us that maybe those changes in methylation patterns could allow these oysters to maintain that egg production in these really stressful conditions. Because we didn't see any difference in gonad growth between our oysters in those low pH and ambient pH conditions. So maybe changing those methylation patterns allowed these oysters in low pH to stay on the same track as those other oysters in ambient pH conditions. Those genes involved in gonad development were sometimes also involved in larvae or baby oyster development. We saw lots of genes with different methylation patterns that are also generally important in early embryos and larvae, so the very beginning of development. And if those methylation patterns are inherited by the larvae, right? So if there are methylation patterns in genes in eggs, right? And then those eggs get fertilized and develop into embryos and larvae. Through that process, these different methylation patterns could be inherited. And if they are, maybe methylation from adults could influence larvae and influence their development processes. So that broad concept there is called carryover effect, right? This idea that adult exposure to a stressor can influence how the offspring do. And if we think about these, if we step away from DNA methylation for a moment and just think about these broadly, we can say that parental exposure can be either helpful or harmful for larvae, right? So helpful parental exposure to ocean acidification means that maybe the larvae are going to be bigger, they're going to be faster growing, or they're going to have maybe better survival. And this has been seen in Sydney rock oysters, so this oyster species in Australia. So when these adults were exposed to ocean acidification conditions, their offspring were much faster growing in those same ocean acidification conditions than offspring from parents who didn't get exposed to ocean acidification. Right. On the other hand, parental exposure could be harmful. And this is what's called a negative carryover effect. Resultant larvae could be smaller, slower growing, or have much worse survival. And this is something that's been seen in a lot of shellfish species on the East Coast, where some Atlantic hard clams and bay scallops, if they were exposed to low pH conditions, their larvae actually developed a lot slower in those same low pH conditions. So we were interested in that as well. So that same experiment I just told you about with methylation, we took some of the oysters and sampled those gonads, and I talked to you about the methylation data. But any oysters that were remaining, we put in ambient pH conditions. So these are non-stressful conditions for 12 weeks. And then we spawned them. So we cut them open and we took their eggs and their sperm and we mixed them in little beakers, and we hoped that we had larvae that would grow. So after we spawned these oysters, we measured the survival of the larvae 18 hours after we fertilized the eggs. And so I'm going to show you a box plot right now. I have the proportion of larvae that were alive 18 hours after fertilization on the y-axis. And then I have female treatment, so were moms exposed to ambient or non-stressful conditions or low pH, stressful conditions. And what we found was this maternal effect on larval survival where if moms were in those stressful low pH conditions, then the larvae had much poorer survival 18 hours after fertilization. And this is actually kind of interesting because one, our larvae were exposed to only ambient conditions. They weren't in these stressful low pH conditions. And two, these female oysters were also kept in ambient pH conditions for 12 weeks. So they had only been stressed out 12 weeks before they were spawned. And you still had this effect. So we saw that this long removed stressor could still impact larval survival and potentially any changes in methylation patterns that the adults experience, right? So if those female oysters were changing their methylation patterns in stressful pH conditions to maintain egg development, that maybe those changes in methylation patterns were inherited by the larvae and they weren't beneficial for the larvae. So it's a possibility that this is this negative carryover effect that we saw could actually be because of changed methylation patterns. Right? So how do we use this information? There's a real interest in what's called stress hardening in hatcheries, where we can expose oyster adults to these stressful conditions and then spawn them to try and see if that will maybe help the larvae if they were exposed to those same conditions. But clearly it's important when that stress hardening occurs, because if we expose those adults to stress 12 weeks before they spawned, and that didn't really give us an outcome that we wanted. So knowing the history of those adults that were spawning and being really careful with when they're exposed to stress might be really important if we're trying to maintain oyster yields in these stressful conditions. So overall, I hope, one, you learned a little bit about how we can use molecular techniques to investigate how oysters respond to ocean acidification. And two, thank you. And I'm here, I think, for quite a bit to answer any questions you may have on any of this work. Thank you. Sorry, I got my booster shot yesterday. My brain's a little fuzzy, so hopefully I did my best to kind of explain everything.
Hi, Yaaminin, can you hear me?
Yes.
Sorry, the power went out completely in our building, so I lost the last part of your talk. I assume it continued for you and everybody else in the room.
Oh, I assume so.
Sorry, it looks like at least one chunk of campus is out of power suddenly. So I apologize. Hopefully you weren't waiting too long.
No, I just finished.
Okay. So let's see if Jim from his phone can actually operate the chat. This will be really entertaining. Let's see. Okay. Let's see. So we do have a question. Have hatcheries taken any kind of action to respond to ocean acidification?
Yeah, that's a great question. Hatcheries have taken a lot of action to respond to ocean acidification. And there are a couple of things that hatcheries can do. Some hatcheries can actually control the water chemistry of the water in their hatcheries. So a lot of hatcheries are set up right next to like water bodies where they pump in the water for their hatchery. And if you're a bigger hatchery, a lot of times they can control how acidic those water conditions are. So they control the pH of the water. You can control water temperature. You can add extra oxygen to the water if you need to. So you can control the conditions of the water when it's in the hatchery. However, that gets a little complicated because part of the hatchery cycle involves you raise the babies in the hatchery, but after they get to a certain size, you put them out on the tideland. So you put them in the natural environment. And then you can't really control what happens in nature, right? So they may be exposed to something stressful in those environments. That's kind of one example of how hatcheries have tried to take control of climate change situations. There's also a lot of interest in selective breeding of oysters. So breeding oysters that are going to be resilient to stressors like disease and ocean acidification.
Let's see. Folks, if you have your questions, it may be because I got kicked off that I can't see other chat questions. So if you want to re-chat your questions, wonderful.
I think I just got a question directly messaged to me. So yeah, the question was, can you repeat or clarify what, if any, effect eelgrass had on oysters? So I'll start with that part first. Being in eelgrass actually didn't have any effect on oysters, right? We thought that, right, eelgrass, as they photosynthesize, can actually buffer that, buffer pH conditions next to them. So we thought that putting oysters in these kind of buffered conditions, we thought we would see that impact at a protein level. And we didn't see that impact at a protein level. We had collaborators who looked at oyster growth inside and outside eelgrass beds, and they didn't see any impact on oyster growth either. So this kind of idea that we had that eelgrass, being in eelgrass, would be beneficial for the oysters, we didn't see it in Washington. However, there are researchers down in California who've also done a similar study, not looking at protein, but just shellfish growth. And they found that in California, for oysters down there, eelgrass presence does help. So it's kind of an interesting concept where maybe in some locations it'll be helpful, in some locations it isn't helpful. And I think a lot of that is contingent on the oysters themselves. If they are, you know, like I mentioned earlier, some oysters are just hardier than others. And so it'll be interesting to compare that. The second part of this question was, it sounds like pH is potentially more problematic than temperature increase or volatility. Yes and no. So if we're looking at stressors in isolation, like if we're looking at only temperature or only acidification, right, we're going to see some very different impacts than if we're looking at things like pH, temperature, dissolved oxygen, and salinity all at the same time. So I think what happened with our work is that if we're looking at all of those things at once, temperature maybe seems to be more important. And there's some there are lots of other studies that have tried to look at stressors occurring together. And sometimes temperature is one that kind of comes out as being more important, quote unquote, or like more problematic. But I think that it's kind of, from a hatchery level, I think that pH is potentially more problematic than temperature increases. If you're trying to control conditions with a hatchery, it's a lot easier to control temperature conditions within a hatchery than it is to control the level of dissolved gases in the water within a hatchery. So I think it just, in terms of what's more problematic, I think that answer, right, is that it depends. It depends on where these oysters are. It depends on what can be controlled in a hatchery, and it depends on, again, the specific oysters themselves. Some lines of oysters are just hardier because they've been selectively bred than others. So it's a very good yet complicated question that people are, I think, still trying to disentangle, because it is complicated because living things are complicated. Oh, okay. I just got a question. So I'll read it from the chat. So let's see. As you know, the Access in STEM program co-sponsored my talk today, and yes, thank you. And we have many of our Access scholars, STEM interested undergrads in the audience today. I would love to know more about what initially sparked your interest in science, and then what supported you in continuing to pursue those interests through undergraduate and graduate school and your current position. Were there any particular experiences, influential mentors that impacted your choices? That is a great question. So I'll kind of start from the beginning. Jim mentioned in my bio that I grew up in the California Bay Area, and I grew up going to the Monterey Bay Aquarium. And so I would say that originally sparked my interest in science, and marine science in general is just seeing marine science, which I realize is such a privilege because not everyone grows up near a wild space or near like science museums or aquariums. So it's definitely something I was very lucky to experience growing up. And then I ended up volunteering at the Monterey Bay Aquarium in high school, which is where I think first started realizing that people actually were marine scientists for a profession. I worked with some marine science educators, as well as people who worked in the aquarium themselves, like aquarium scientists, aquarium technicians, people who did husbandry or like took care of the animals. And I was like, oh, like this is an actual job people actually go to school for. And I had a really great mentor through my volunteer program that, you know, I mentioned I was interested in kind of being a marine scientist, a marine biologist. And she was like, that's great, take as many classes as you can in college, but don't major in marine science, which is kind of interesting advice that I got. What she said was, you know, major in biology because you're still so young and you don't know what you might want to do. And having a general biology education is potentially going to be more applicable for you. Like you might learn more general things, be able to work in different capacities. And if you change your mind, you still have a great degree that you can do something with. And if you don't change your mind, that's totally fine. So I went to undergrad and kind of kept that advice in my head. I took general biology classes and tried to get a lot of paid research experiences. So I participated in a couple of research experience for undergraduates programs, which are summer research experiences that are funded by NSF, meaning that for 8 to 10 weeks, you get to do marine science research and you are paid to do so, which is a really great program. There are locations all over the country. And if you've never heard of this program before and you're interested in STEM in general, not just marine science, but like any kind of STEM field, there's probably an RAU for what you're interested in, and you should completely check it out because it is a great way to get like that research, hands-on research experience, because you kind of work with a graduate student or professor mentor to develop a project, and then you carry out that project, write up the results, and present the findings all as part of your RU. So you learn a lot and you get paid to do so, which is very important in this field. So I would say those experiences in particular really kind of showed me that I could do marine science research, and it's something I was interested in. And then I would say going into graduate school, I was very lucky to have the advisor that I did. I think graduate school is a really difficult time because you're like putting a lot of yourself, I think, into your research. And so it's great to have a supportive advisor and lab group that is going to help you thrive and help you become the best scientist you want to be. And so I was very lucky to have that. And I had a great cohort of other graduate students that, you know, we weren't a very competitive program. We were all just trying to make sure that we each succeeded in our own ways. And so I think like that community in itself was very influential for me and impacted my choices. Yeah, I could go on at like for a lot of these things. And, you know, I think my email is somewhere. So if you have specific questions about, you know, pathways to marine science or STEM in general, and like, you know, what graduate school is like, or what RUs are like, I'm more than happy to answer those offline too.
Cool. So now that I've unmuted myself, it looks like we have another question. Do you have thoughts on key steps to restore local fisheries, including and beyond oysters in the South Puget Sound region?
Yeah, so that is a great question. And I have thoughts. I will caveat this by saying my expertise isn't in fisheries management and restoration, really. I come at it from more of a, how can I generate the information that maybe these resource managers and and various entities need to make the decisions that they need to make, right? Like I'm kind of on that other side. I will say though, the biggest thing, in my opinion, for successful restoration is making sure that it is equitable and inclusive restoration, right? So involving all of the various community groups that would be impacted by any of these decisions, right? So like bringing not just like state agencies, but like bringing the tribes to the table, bringing various communities to the table if they're going to be impacted by those decisions, and trying to and taking their wants and needs into account very earnestly to ensure that, that when a system or species is restored, it's not just restored because it's not just restored because it should be restored, it's restored because I'm not saying this correctly. It's like not just restored in a way that maybe is purely based on biological science and numbers. It needs to feel good to the people who use whatever fishery resources exist. And there definitely is a balance to all that, and that balance can be achieved. And I think just the first step is making sure everyone is at the table and everyone is listened to equally, not just at the table and can't talk.
I had a question for you. Did you, kind of thinking about how you would apply your work to management, did you get a chance to look for your markers in natural populations?
Yeah, that's a great question. And I haven't really had the chance to be able to look for those markers in natural populations. I think the closest I got was that first protein experiment I talked to y'all about, where we put these oysters in those kind of like naturally varying environments. I think that there's still a little bit of ways to go when it comes to translating these like really intricate molecular questions to usable information. Like I think the goal is to get to look at the molecular information, figure out what it tells us about like if you can take the molecular information and translate into like, okay, like this species will be able to tolerate kind of like this range of things, and then use that information in mathematical models that are used to kind of predict how species will react to changing conditions, or how many larvae are we going to get in Puget Sound this year? Like, what is the hatchery reproduction going to be like? If we can, I think like that is the goal. And I think there's still a little ways to go to get this kind of like really complex molecular information down to a couple numbers to be able to do that. But I know that, like it's certainly being worked on, and it's certainly like an end goal. But step one, right, is kind of figuring out what's going on, and then we can kind of think broadly about like, okay, like, yeah, at least like, with the work that I do, it's not just, so far it's been like, okay, so like if your broodstock have experienced like really stressful conditions, like even like 12 weeks prior, like that's maybe not going to work out well, or work out as ideally as it could. So it's a start, but it definitely has some room for growth, I would say.
I don't see any other questions, but maybe one last one for you. So how did your work that you did at UW take you to what you're doing at Hui now?
Yeah, that's a great question. So a lot of times after you get a PhD, some people go straight into jobs like research, whatever those jobs may be. And sometimes you do what's called a postdoc, which is, I think, that the most equivalent thing that may be familiar to some people is like the idea of like a medical resident. Like you finished medical school, but you're not a real doctor yet, and you're kind of this medical resident, and you're getting additional training. That's kind of what a postdoctoral research position is you've just finished your PhD, you got really good at one specific thing, but you maybe want to learn a couple more things or apply the work that you did to something different before you apply to jobs to make you a little bit more competitive. And there's various different ways to be a postdoc. There are postdocs that work with federal agencies, postdocs that work with nonprofits, and then postdocs at research institutions like me. So I got really focused on kind of DNA methylation and oysters for my PhD, and I wanted to still think about omics and how marine invertebrates respond to climate change, but I wanted to study some different organisms because there's very different contexts. So that's kind of how I ended up at Woods Hole and doing the work that I do now. Is I wanted to learn more, and I wanted to learn some new things and think about how I could take the thing that I got good at for 5 years and apply it to something else, so I could, make more of an impact with what I do.
That's wonderful. And thank you. Sorry for the snafu with, it turns out the entire university campus is without power. So I'm glad I could rejoin you on my phone. And I think that might be why you lost quite a few people all at once, as everybody in the university suddenly couldn't get on. But
Oh, wow.
really appreciate it. The recording is still good, so everybody will be able to see this afterwards. I really appreciate your time. Thank you everybody for coming and checking out the talk today. And Yaaminin, best of luck with everything that you are doing at Hui, and we'd love to hear more about what you do in the future.
Yeah, thank you.
Thank you.
And thanks
Take care,
everyone
everybody.
for having me.
Yeah.
Bye.
Bye.